Good evening, and welcome to Pacific Science Center's virtual Science in the City talk this evening. My name is Zita, and I will be your host tonight. And I want to thank you all so much for joining us for this virtual experience. Now, even though this is a virtual Science in the City presentation, it is still interactive. So on your YouTube page that you're watching this, you can see the chat box. And in that chat box, you can send us any questions you have about tonight's presentation, any questions you have for our speaker. And after the initial presentation, we'll be asking your questions of our special guest speaker. But that interactivity relies on you. So go ahead and get started by saying hello tonight in the chat box. Let us know where you're watching from and let us know how many people you're watching with tonight. Before we get started for tonight's presentation, I just want to share a little bit of information for you. We've uploaded some of our past Science in the City talks, in fact, all of our past Science in the City talks, to our YouTube page. There you can watch the Mars surface exploration, looking at the past, present, and future of Mars exploration, just in time for the landing of the rover in just a few short months that launched last month by NASA. You can learn more about the Climate Pledge Arena that is right there in the Seattle Center and learn the science behind that arena that's being built. All those talks and many more can be found on our YouTube page. Educational programming like this tonight is thanks in part to the generous support of donors and potentially donors like you. In the faces of COVID-19 and the challenge of climate change, science and an informed public are absolutely essential. And if you're able, we'd like to suggest a $10 donation for tonight's event to help us ensure that curiosity never closes. For more information about donating to Pacific Science Center, you can visit pacsci.org/support. And now for tonight's event. You know, this year it feels like we've all become armchair virus experts. And if you're anything like me, that focus has been on how to prevent the transmission of an existing virus outbreak. But scientists like our guest tonight are hard at work understanding these types of viruses before they become a human pandemic. And tonight we're going to learn about the history of specific types of coronaviruses and looking into the ability of these viruses to jump across species lines and how scientists are working to be able to predict these types of viruses and outbreaks in the future. So joining us tonight is Dr. Michael Letko. He's a virologist from the Washington State University Paul G. Allen School for Global Animal Health, and his research focuses on cross-species interactions. Michael has a BA in molecular biology and a PhD in molecular virology, and he's developed a deep appreciation of how viral diversity at the smallest scales can dramatically influence how viruses and their hosts co-evolve. Now, through his research, he has sampled African fruit bats in the Republic of Congo, and he has screened dromedary camels in Jordan. Now, Dr. Letko joined the faculty at the Paul G. Allen School at WSU in just this summer, the summer of 2020, and has opened the Laboratory of Functional Viramics, the Laboratory of Functional Viramics. And that work is centered on the concept of testing groups that are related novel viruses and how these can cross species barriers. The hope is, and the goal is that one day this work will help us to respond to and maybe even prevent the next emerging pathogen. Dr. Letko, welcome, and thank you so much for joining us this evening.
Great, thanks for having me, and thank you for that really wonderful introduction.
Thank you.
Okay, let's go ahead and get the screen sharing going.
Sounds great. And again, remember everybody, as you're watching through this presentation, you can put any comments, questions that you have in the chat box on YouTube, and we'll be asking our guest those questions after his presentation.
Great, thanks. All right, so thanks for tuning in tonight. So today I'm going to talk a little bit about the work that I do on coronaviruses and looking at how these viruses transmit across species barriers. So I'm interested in coronaviruses and this question in particular because really over the past 17 years, coronaviruses have become, they've really shown their threat to global health. And this really started in 2003 with SARS coronavirus emerging in China. It quickly spread to about 8,000 cases before it was tamped down. And basically shortly after the virus emerged, groups started trying to find out where it had come from, and it was quickly traced back to the palm civet in open air markets and raccoon dogs in open air markets in China. And then several groups quickly started looking into other wildlife around the region, and they found not exactly SARS itself, but several related viruses in horseshoe bats, in the Rhinolophus species of bats. And so this question came out that, okay, it looked like SARS clearly had gone through some intermediate host and it jumped to humans, but it looked like it even maybe a possibility that viruses could originate in bats and directly transmit to people. But regardless, we see this kind of theme emerging of one virus having to encounter and replicate in multiple hosts. And since the discovery of those bat viruses early on in 2005, now many groups have found hundreds of other related viruses to the SARS family. We call them sarbecoviruses. In 2012, then we had the emergence of the Middle East respiratory syndrome coronavirus. Unlike SARS, this one is still an ongoing problem in the Middle East. And it's because this virus transmits to humans from dromedary camels. And unfortunately, unlike SARS-1, this one seems to continuously transmit. So it's been very tough to eradicate it completely in the region because it's constantly coming back into the human population from camels. And similar to what we've seen with the other virus, with SARS, several groups have now found multiple related viruses to the MERS virus in bats, and actually several other species. But we call this group of coronaviruses the Mirbeco family, and there are actually now at least several thousand sequences in the literature for these viruses. Something more for the vet-oriented crowd, there was the swine acute diarrhea syndrome coronavirus that emerged only a couple years ago in China. This transmitted directly from horseshoe bats to pigs, and it had a very high mortality and impact on the pig industry. And then, of course, we have the virus that we're now all, unfortunately, way too familiar with, the SARS coronavirus 2, which last I checked was about 27 million cases and over almost close to 1 million deaths. This is unfortunately full of a lot of unknowns. So we're not really sure if there is an intermediate host that might be giving us SARS-2. We have some pieces of the puzzle like we have for the other viruses. So already we've now found several more closely related sarbecoviruses to SARS-2 circulating in bats. So whether or not it goes through an intermediate host or jumps directly is still yet to be determined. But then just to kind of remind you, again, SARS-2 is yet another member of the sarbeco family of coronaviruses. So what we're learning now through all of these projects where groups are going out into the wild and sampling bats looking for viruses is that we're finding viruses everywhere. We call this collectively the virome, refers to all the viruses circulating in all the animals around the world. And so this is kind of where we're at. We're at the virus discovery stage, and as a result, we're now accumulating a lot of sequence data. So using next generation sequencing technologies, we're able to actually pull out the genome for all of these viruses in a single sample. And out of all of these viruses in the virome, hundreds are sarbecoviruses, thousands are mirbecoviruses. But unfortunately, most of these viruses have never been isolated or worked with in the lab. So the really big questions that a lot of the science community is interested in, such as can these viruses transmit to human and cause a problem, remain basically unknown because we haven't actually had a chance to work with them in the laboratory yet. And that's where this work and the work that I'm interested in pursuing now is really falling underneath this kind of second round of research where we're now taking all of the data that we've accumulated on the sequences and trying to develop new tools and approaches that will allow us to study these viruses in the lab so that we can answer these questions more accurately, whether or not a virus can infect a human cell, for example. So up until this point, so over the past 17 years since SARS was discovered and all of those other viruses were found in various animals, we have studied, and by we, I mean the scientific community has collectively studied 9 sarbecoviruses and just 4 of the sarbecoviruses. So coming to the sarbecoviruses, because this is where the rest of the project really starts to move in the direction. So in order for any of these viruses to infect a new host, they need to first be able to infect the cells of the host. And in general, a lot of viruses work like this, but the way the coronaviruses do this in particular is through a piece of the actual virus itself called the spike protein. And this is a little protruding part of the virus that sticks on the outside, and it actually physically binds a protein on the outside of host cells, and that leads to this virus being internalized, and then it can then undergo replication within the host. But really, in order for this to take place at all, for any of this replication and infection to take place, it really comes down to this very small interaction between the receptor binding domain and of the spike protein and the host receptor protein. So when these two pieces interact, then the virus is able to internalize into the host cell and infect. If this interaction doesn't happen, then the virus is unable to infect. We call them coronaviruses and we call them spike proteins because when we look under the microscope, I guess the folks who look at these types of images all day and I guess who don't see very much of the sun are reminded of the sun from this, and they start to think that it looks like the corona around the sun and that these little fuzzy things on the outside of these virus particles actually look like spikes. Taking a closer look at what this actually looks like at the very smallest molecular level through more advanced structural techniques, this is the spike protein on the outside of the viral particle. And then this is the host protein. In this case, for SARS virus, it's called ACE2 is the name of the gene and the protein that the virus uses to infect your cells. And then when we're looking, really coming back to this little diagram, the receptor binding domain is just a very small region of this spike protein that actually mediates this whole interaction. This whole initial infection starts kind of at this level. So that got me thinking about how can we study this receptor binding domain from all of these other viruses. We know the sequence for the receptor binding domain for a lot of these viruses. We just haven't ever isolated them. So how can we actually start to get at studying these things that we've never actually pulled out of the samples? And so what I'm showing here now is a schematic representation of the gene encoding for this whole spike protein. So the virus has a gene and we're just looking at it now, reading it from left to right, kind of as the genes are written out. And so this receptor binding domain is actually, it's a very small stretch of the entire gene that actually encodes for this little bit of the protein that then interacts with your cell. And so what I've done is I've made a version of this SARS gene that allows me to delete its receptor binding domain and replace it with the sequence for the receptor binding domain from any of the sequences in the literature. And this is a really cheap and time-effective approach. So before this method, groups would either have to isolate the virus, which almost never happens, or they could try to recover the virus through kind of through a similar method. But before now, it used to take about $15,000 and several months to have the whole genome synthesized. Now we can just go after this very small region to get at this question. Okay, so what does this look like? Basically what we do is we take this gene in the lab, we can then have this little fragment synthesized, we then replace it into the SARS spike. We can then put this into cells, which then produce a safe fluorescent particle. So what we do is we give these cells the weird chimeric gene that we've made, and then they end up making these little reporter particles that look like a virus for the most part. They have on the outside, just like SARS, they have this protein sticking out with our, chimeric, whatever this little receptor binding domain is sticking out. And then when they actually bind onto a new cell and they undergo that process of trying to infect the cell, rather than produce new virus, they just turn the cell a fluorescent green color that we can really easily see under the microscope. So then we can see if the particles turn the cells green, that's indicative that they were able to enter. And that tells us a lot about this little bitty piece that we're looking at. It tells us whether or not that part of the virus can interact with human cells. So if we look at all the coronaviruses that are in the literature, there are thousands of sequences. If we look at just the sarbeco family, like I said before, there's several hundred, although this number is now going up. And of those several hundred, if we look at the receptor binding domain, while all of the viruses are different throughout, really there's only 29 variants of the receptor binding domain. So we had them all made, and then I made those little fluorescent virus particles with them. And this is basically the family of, or the full set that we're seeing. So now I'm going to show data where we're looking at the entry of these various particles and how well they can get into primate cells. Let me see if I can quickly, there we go. All right, so what we see here is actually, I'll just walk you through the controls first. So in the absence, if we make these particles, these little reporter particles without any spike protein on the outside, so they're bald, they're not able to get into cells. They don't have any viral machinery for that. And so we don't get any signal. So this is slammed all the way to the left side of the graph. But for example, SARS coronavirus, which we know can infect primate cells very well, these our signal now is much increased compared to the no spike signal. So from this, we can say these little particles that have this bit of SARS on the outside are actually able to infect and get into these primate cells. And what we see is actually a number of these other sarbecoviruses, and some of these have been studied before, but a number of these sarbecoviruses are actually able to get into these primate cells. And then surprisingly, and this is what we see in this clade 2 group of viruses, is several of these are actually able to infect cells. And so up until this point, like I said, researchers had only studied a handful of this set of viruses in general. They had studied maybe WIV1, WIV1 and SHC, and then they had studied RP3, and that was basically it. So from this, before now, several groups had pretty much determined that really it was some of these clade 1 viruses can infect, but none of these other viruses can. And now what we're seeing is when we test actually more viruses, we see quite a different story come out. We see the same effect for human cells. So this is with primate cells that I'm showing the data for, but I also did the same experiment on several different human cell lines, and we see the same phenotype. But when I start to look at what the actual receptor protein is, what is the part of the cell these viruses are actually coming into contact with to get into the cell, then the story starts to change. So SARS-1 and all of its genetic relatives here in red, these clade 1 viruses, all use this receptor called ACE2. So if I just have cells that only express ACE2, then we only get entry of the clade 1 viruses. But all of these viruses over here that were able to infect primate cells are not able to infect cells that just have the human receptor. So they're using some different receptor here to get into these cells. If we take a look now at bat cells, this is a kind of a random bat cell line that I had in the lab. We actually now get kind of the inverse of the picture. So now we don't see any entry of these clade 1 viruses, and that's now been shown that these cells that we're studying don't have an appropriate receptor. They just don't, they're missing it, but they do have a receptor, whatever that is, for these clade 2 viruses. Okay, so what do we take home from really all of this data? So what we're learning now is that other bat coronaviruses can infect human cells. So out of all the sarbecoviruses that we've looked at, several of them seem to have what we could say now is a higher risk of spillover because we can see compatibility at the very basic level with these viruses and cells and human cells. But we know that they use a very different receptor from SARS. So unlike SARS, which uses ACE2, these other viruses are using something different. And this has pretty important implications for transmission and spread and pathogenesis. So these receptors are expressed in different tissues. So you can imagine that if it's using a different receptor, these viruses would be able to infect a different tissue which could cause a different disease. It can change how they transmit. And of course, it'll change how severe patient outcome would be if they're infected. So we also look at the conservation of these receptors. So what do I mean by that? Well, as I said before, this is kind of an arbitrary bat cell line. Artibius jamaicensis is actually a Jamaican fruit bat that we find in Central South America and the Caribbean. But the clade 2 viruses were all isolated from Rhinolophus sinicus bats in China. So whatever the receptor is that these viruses are using, we can say it's the same between human and primate Rhinolophus sinicus bats and this other bat species, Artibius jamaicensis, all the way on the other side of the world. So we thought we were basically done with the work here, and then started writing everything up in December last year, and then January came by and changed the story. So on January 8th, we started to get reports of a new coronavirus emerging in China. On the 10th, the genome was published online, and it was very quickly apparent that this was another sarbecovirus. So that Monday, I designed the gene fragment for this platform. So keep in mind, because we don't have to wait for the virus to be isolated and we're using synthetic biology, we can get this these reagents made much quicker. The fragment showed up in our lab 3 days later, and then that kicked off about a week-long series of protocols and procedures to get to what the receptor might be for this new virus that's coming out. And so similar to the data that I've already shown you, in 12 days, we were able to go from a sequence on the web page to having laboratory data showing this virus was using ACE2, just like SARS-1. And so this is kind of like the experiments I've shown before, where we're giving now cells various receptors for different coronaviruses, and then we're seeing if the virus particles can infect these cells. And so it's pretty clear that basically only ACE2 allows entry of both the SARS-2 receptor binding domain, but also some of these other clade 1s. And these other receptors for other coronaviruses like MERS and the common cold do not give us any entry. Okay, so why is that important? Well, basically at the time that this was coming out, there was very little information. This was in January. There was basically no information on the virus in the lab. And so there was speculation as to whether or not this virus would use ACE2. Several groups were able to conclude, maybe a little too quickly, that that SARS-2 is using ACE2 less efficiently than SARS-1. And so they were saying that, oh, SARS-2 won't be a big deal. And then we had data kind of saying quite clearly that this virus was able to infect cells with a human receptor better than the original SARS-1 virus, which of course had some implications for transmission now. But also this was the first set of laboratory reagents for this virus. So like I said, we moved right from the sequence online to having reagents in the lab in 12 days. And because of that, we were in a place to really quickly distribute these reagents around the world to labs studying vaccine development and serology assays. So what do we know about these sarbecoviruses that we've looked at? So clade 1 and 2, the two big and biggest groups of viruses that we've looked at within the sarbeco family, mostly were found in Rhinolophus sinicus bats in China. The one clade 3 virus that we studied that there's a sequence for online was actually identified in another Rhinolophus species in Bulgaria. And although we don't have the full viral genomes for these and weren't able to study them, we do have viral sequences for a number of other countries and other Rhinolophus species. So it's very likely that the sarbecoviral family is actually much more widespread than we anticipate. So because we know that they have a wide distribution and that these viruses can, or at least some of them can infect human cells, we're also seeing that they're discovered pretty frequently. So now in light of SARS-2, several groups are hitting back into the field to look for more of these viruses, and there really is no shortage of them. We do find them in a lot of places, and we're finding them now in new species. So for example, pangolins might be a species that we're all oddly familiar with because of the news and SARS-2. So the more we look, it's very likely that the more we find. But now that we have some tools in place, and now that we're starting to expand the Sarbeco database again, it's clear there's a renewed need for surveillance of these naturally circulating viruses. If we can look at the viruses that we find on a more continuous basis, then maybe we can try to take steps to mitigate and prepare for potential outbreaks earlier. So kind of as a proof of concept, as we were working through the first project and studying the published set of sarbecoviruses, we paired up with Simon Anthony and his student Heather Wells at Columbia University. Simon's affiliated with the EcoHealth Alliance, which is affiliated and a partner with USAID's PREDICT program. PREDICT has also been in the news lately because they have a sarbecovirus and, well, just a viral discovery program in general, and have been aiding in the discovery of sarbecoviruses, not only in China, but elsewhere. So they had found some sequences from Uganda and Rwanda, which we just quickly tested for their ability to use the human receptor as a proof of principle for this type of surveillance work that can happen. And so just like what I've shown before, I took the SARS-1 spike and I replaced its receptor binding domain with the sequence from these other viruses. These viruses are clade 3. So this is kind of exciting because, like I said before, we're only able to study the one clade 3 virus that's online. And so now with this collaboration, we were able to study a few more and get a better idea for what's going on. Unfortunately, well, I guess fortunately, actually, we see that when we give cells the receptor, the human receptor ACE2, we only get entry with SARS-1. So these other viruses, these other clade 3 viruses are not getting us any entry there. So just to keep in mind, though, we've only found 3 of these clade 3 viruses, and it seems to be that the more closely we look, the more often we find them. But we know that if we look at clade 2, for example, some of the clade 2, not all, but some of them were actually able to infect human cells and use a human receptor. So I think that this really argues for the need for more screening because it may be that as we find more clade 3, just like clade 2, we'll find a few more viruses that are capable of infecting. So to that end, really been trying to get my foot in the door with some of the amazing work that they're doing at the Paul Allen School and some of the international work that they have been running with their collaborators. In particular, we've now submitted some research proposals to carry on with this sarbecovirus surveillance, both in Kenya and in the Paul Allen School's Tanzanian program. And I'll just leave you with this. So what do we do with this information? All right, so we can sit here and test all of these viruses, but why do we care to have this? What does it actually get us? So I think the first and some of the most immediate implications, and ones that I'm actually in the process now of pursuing, would be to go after broadly protective sarbecovirus vaccines. So rather than go after one specific virus like SARS-2, we could go perhaps more broad, like a clade 1 virus or a clade 2 broadly protective virus. So trying to use the information that we have now to do more rational vaccine design. As we have started to learn what viruses are carried by what species and what species of virus have the most risk, we can start to then come up with ways to reduce contact risk, contact with some of these higher risk species. And I mean, it's good. We look at bats a lot, but it's important to keep our focus not just on bats, but also on the domestic animals that we interact with and the wildlife that come through our trade, because that's really where we see a lot of the coronavirus transmitting, at least for humans up to this point. It seems to be that it's not necessarily always coming to us from bats, but it comes to us from something that we're more closely associated with. for example, dromedary camels in the Middle East, or animals in the wildlife trade in China. As we have started to now get a better picture of how these other viruses function, I think we can start to use this as a jumping off point for looking at how these other viruses actually infect. So we know about these clade 1 viruses. They look like SARS. They use receptors found in the respiratory tract and the gastrointestinal tract. But what about these other viruses that we see infecting cells? How are they getting in? And the more we learn about them, the less of a surprise these viruses will be if we ever see them come up again. And then finally, can we apply what we've learned with the Sarbeco family of viruses to the Mirbeco family of viruses? Which, like I said before, there's thousands more in the database. So if we can really start to kind of expand into other viruses, then we can build up a database of some of these risk factors, and it really gives us a little bit more predictive power. And with that, I'll thank everybody at the Paw Island School for Global Animal Health and our collaborators, and I will take your questions.
Excellent. Thank you so much for that overview. As he said, we will take any questions and comments that you have. So go ahead and put them right there in the chat function of this YouTube video. You covered a lot of different information. And so I'd love to actually start a little bit at the end. And you were talking about the different animals that they can jump species. And you commented that it's not always bats. It can be domestic animals and it can be animals from the wildlife trade. Why is it, though, that we seem to jump to bats so easily as part of that thought?
Well, I think, bats are, we've looked at them maybe the most out of all of the animals that we have in nature that we look for viruses. It tends to be bats and rodents just because they're so incredibly abundant. Bats are, there's so many of them. There's so many different species and such a breadth of diversity. So there's a lot to study there, I think, really is part of why we look at them so much. And it's I think because we know they carry so many viruses, it's an easier place to start than to pick some, let's say, ground shrew and see what viruses it might have.
Is it possible, are these types of viruses going to be found across the animal kingdom, or are they more prone to be in certain types of animals and not in others?
Well, with the sarbecoviruses, that's maybe a little bit less known. So we know that they're in a lot of bats, and we know we have some intermediate species ideas, like we know that pangolins maybe seem to be able to carry sarbecoviruses. We know that palm civets and some of these smaller weasel type creatures can carry these viruses. But I think it's really just kind of a sampling bias, though. So people have mostly looked at bats, but as we look into other things, we find more. The Mirbecoviruses are a really good example of that. So Mirbecoviruses have been found in not most, but they've been found in lots of different mammalian species.
Excellent. All right, so we've got some great questions coming in from our guests now. Lost Laboratory is asking specifically about the SARS-CoV-2 and asking if there's evidence that it's passed through an intermediary host, or is it pretty clear now that that spillover occurred directly between bat and human?
Yeah, so this is a big question that a lot of groups have been asking. And there's some really nice work, I think, that's coming out now with regard to that. But I think it's largely unknown. There is some genetic data that really does, or some groups are trying to argues for the virus going more direct from bat to human. But again, I think it's a little tough to say if nobody's been able to definitively rule out an intermediate host. Let's try to play it safe that way.
Is that something that is possible to actually rule out, or will it always be a bit of a mystery at this point?
Yeah, I'm not sure. That starts to get a little too macro for me.
Okay. That's totally fair. So we've got another question about all the viruses that you've discussed, or are they all the beta coronaviruses? And do alpha and gamma coronaviruses also pose a threat of zoonosis?
Yeah, so these are all beta coronaviruses, and specifically, most of the work that I showed was all with the the sarbeco family of viruses. So just the viruses related to SARS beta coronavirus. But yeah, for example, the alphas and some of the other coronaviruses, of course, might also be a risk. But we study, I guess, we choose to study the things that are closest to what we know, right? So beta coronaviruses are a great place to start because we just happen to know them, I think, some of the most about them, both structurally and functionally. But then as we learn more about these viruses, we can start to expand into the alphas. But whether or not they propose just as much of a threat, we have not seen a pandemic alpha coronavirus.
Is any guesses as to why that is, or is it just one of those things that we just haven't studied it long enough to be able to rule that out?
Yeah, it's maybe it's perhaps just a studying thing, or just that we haven't found or had the right opportunities for those viruses to transmit. Maybe they can, and we just haven't quite gotten into the habitat of those animals yet, for example.
This might be a really simple question. Are new viruses, I mean, there are viruses that we're going to have in the future that don't exist right now, right? Mm-hmm. So, let's start with that initial, is that a truth? That viruses are growing and changing so that they are new ones?
Well, yeah, I guess that's kind of like, so some of the viruses that we look at, like SARS-2 and SARS and MERS, for example, groups have tried to pinpoint when exactly those transmission events took place. And, we'd say they're relatively contemporary compared to like evolutionary time happening within, I guess, we'd say a contemporary time frame of only a couple hundred years rather than thousands or millennia, for example. Mm-hmm. And some groups will trace that back to, the recombination events that maybe led to those viruses kind of forming newly from existent viruses. But yeah, well, I don't know. We'll leave it at that.
Sounds great.
It's easy to get lost on that
one. So you've been studying viruses for quite a while. What is it like to be studying something that has so quickly become the hot topic in the world, in the news, in people's lives? How's that process been for you just over this past 9 months?
People are more interested to hear about what you do. Well, I mean, only the important parts.
What are some of the most common questions that you're getting from people?
Yeah, I guess the question that I still get is just people wanting to know where it comes from and like, oh, do you think it was made in a lab? And I think, for somebody that's looked at all the other members of these families or of this family, it's pretty clear that they seem to originate in nature, and they circulate naturally. There's more than, it's not just one virus that did it. There's a number of them out there. So, where it comes from is not, it's only so much of a mystery.
It has a recognizable pattern that fits what you've seen very closely in nature.
Right.
Yeah. So thinking about all these viruses that are out there, what kind of surveillance is being done to detect potential future zoonosis?
Yeah, so like I said, especially now with the kind of influx of SARS-2 money in the research community, a lot of groups are now turning the effort again to looking for sarbecoviruses, and really, as the nice thing is, since the first SARS outbreak in 2003, there's been this kind of nice explosion of technological advancements on the sequencing front. So the folks that are doing the virus discovery are able to pull out a lot more data than ever before from their samples using this, some of the same types of reagents, and some of the new technology that's coming out. So I think that, yeah, there are several research groups now continuing to sample wildlife around the world. It's not just EcoHealth Alliance anymore and Predict, or I guess what now Predict has become, but it's also smaller institutions, individual labs around the world, trying to basically piece together more data, more sequencing data at least.
Yeah, that was amazing in your talking about just how quickly in January you were able to get that information and start studying it.
And it's really, I think it's really a tribute to the folks that to provide the sequence data. I mean, the rest of the West was kind of in the dark until the virus sequence comes out, and then we're able to really start hitting the ground developing these reagents. So it was, it's definitely, our speed was only attributable to the speed of the group releasing the genetic data for us.
So the technology, as you've commented, has definitely improved and there's been some great breakthroughs, but I'm sure the technology needs to continue to advance. Where are some of the limits in technology now?
Well, I think, with really the technology is limited at the downstream steps. So we can accumulate all of this sequence data, but how do we study them at a big enough level where we, and get data that we can actually trust, which unfortunately usually comes from just solid laboratory experiments. The world of computation is definitely improving at a crazy fast pace. So it might be that at one point we can just take this sequence data, predict a protein structure for it with a computer, and then do these kinds of interaction analyses all right there on the computer. But as it stands now, I think that technology is still just a little bit, it's getting there and it's improving at a pretty quick pace, but it's still, I think, a ways off from being able to do that. So unfortunately, that means we need these kind of more basic lab tools. And that's kind of where this work fits in. It's just trying to just get it, almost grasping straws, because it is such a difficult problem to say, I want to study a bunch of things that don't grow. A bunch of viruses we can't isolate. That becomes a challenge.
I can imagine so. When you're measuring the ability to infect human cells, are you testing just one type, for example, respiratory tissue? And is it possible that, well, maybe I'll let you ask, it's a two-part question, so let's answer that one first, and then I'll follow up with my second part.
So we're using a number of cell culture tools in the lab. These are I don't know, we call them cell lines. They're just cells that we've been propagating in the laboratory setting for decades. These cells are not maybe the most physiologically relevant. So they're good for those basic questions of, can a virus infect and use this kind of host cell machinery? But the cell that we're looking at in the plastic culture plate actually reflect what happens in your lungs. It's a little bit less translatable there. But with that said, we have looked at cells that are derived from respiratory tissue, and we've looked at cells that are derived from gastrointestinal tissue and liver tissue for humans.
Is it possible that a virus could be able to infect one type of cell, but not another type, making it important that you're testing across the range of human cells?
Mm-hmm. Yeah, of course, definitely. And like I said, these cells that we work with in the lab have been propagated for a while, so they start to do weird things over time, and we like to think that they're all very homogeneous and they behave a certain way, but it is a little bit of wrangling to get consistent results out of these cells.
So how often are you, how long will the study need to go before you feel pretty confident that you've got consistent, repeatable results?
Yeah. So yeah, this was a project that I started in 2018, and that was the whole first year was just developing something that allowed us to quickly replace out and easily replace out that part of the viral protein. And then it was another year of doing all the experiments. And I mean, I think there were something like 120 experiments done for this project over the course of 2 years. So it really does, it ramps up, and you get used to seeing this kind of data over and over and over again before you want to tell it to anybody.
I'm imagining right now just spreadsheets upon spreadsheets that are pretty huge.
Yeah, it's a lot of screens.
Okay. Yeah, I'm glad that the reality fits what I'm imagining the life is for a molecular virologist. What is it about these viruses, and specifically or viruses in general, that makes them so infectious? Is it structural? Is it due to the conditions of transmission, or are there other factors that are coming into play?
So many factors, I think, contribute to that. And that's something that, especially now that people are really, the whole research community is tuning into SARS-2. And so we're learning even more about, for example, how respiratory viruses will transmit. But I don't think we could say that necessarily all viruses have equivalent infectivity. There's a number of viruses that seemingly actually are fairly difficult to contract compared to other viruses. And one way that we measure that in the virology world is through the R0 value. So talking about how many new cases one virus can bring about. And so, not all viruses have equivalent R0s. They're actually quite ranging. So, I don't know if we can say blanket what makes one virus more infectious, but it does really come down to the genetic level and to a lot of these very small details that we're looking at. If we make that protein a little bit stickier through some mutations, then it might be able to infect lung cells better. Unfortunately, maybe then if it sticks to lung cells too good, though, then maybe the virus doesn't break off of the cell easily enough and isn't able to infect a new person or another cell. So all of these factors kind of balance out, and it's tough to say really one particular component that gives a virus more transmissibility over another.
And there's got to be some challenges in studying that, given that it's not paused. This virus in the world is mutating, is evolving, is changing in real time as you're studying it.
Mm-hmm. What's nice about this one, and really the coronavirus, is they don't seem to move at the same pace that we see in other viruses like HIV or influenza, for example. Influenza changes so quickly, we need a new vaccine every year. But with this virus and with the coronaviruses, they have some limitations on how much they seem to be able to rapidly accumulate large numbers of mutations. So by and large, what we see kind of in the sequence population around the globe, yes, we see 1 or 2 mutations that seem to, fluctuate through a population, but we don't see any mutations coming up that right now, at least, that seem to be interfering with the effectiveness of a vaccine, for example.
What is it about this particular virus that doesn't mutate as much as, say, some of the other ones like influenza?
Yeah, so that comes down to like the size of these viruses. So influenza is a pretty small virus. Its genome is it's much smaller than the genome for a coronavirus. And so that means that in order for the coronavirus to replicate, it can't make those kinds of mistakes that flu can get away with, because if flu makes one mistake every 10,000 letters it copies, it's going to get 1 or 2 mutations. But if these coronaviruses start introducing a mutation at that frequency, it will accumulate potentially too many. And so these coronaviruses have a proofreading function and a little bit of its enzyme that actually ensures that it copies its genome a little bit better than other viruses, for example. So it's in its best interest not to mutate, so much so that it's not stable.
Because if it's not stable, then it's not going to infect, it's not going to be able to survive.
Yeah, if you accumulate too many mutations, just like what we see in humans, if you get too many mutations in a cell or in a living thing, it doesn't usually go well for it.
I haven't often over the past several months heard the sentence, "the good thing about this virus is," But that gives me a little bit of hope. You were talking about the size of viruses. How small is a virus? How are you actually seeing these?
Yeah, so the work that I do, we don't really see the viruses directly. We don't see these proteins directly. We have a lot of more like indeterminate or kind of indirect ways of looking at them. So like I said, when I wanted to study how this protein interacts with a human cell, the way we study that is not to actually look at the proteins, but is to come up with an experiment that lets us see if that little bit of the protein is behaving how we think it would. And we can test all kinds of parameters to make sure that it's where it should be and it is the right size and all of that. But we can do all of that without ever actually physically looking at it because they are so incredibly small, smaller than the wavelength of visible light, so that they don't have color or, the only way we can look at it is with electrons, electron microscopes, and X-rays. So something much, much smaller than just visible light, even.
Okay, I can see how that can, looking at more like the results or the impact on something else for something that just isn't visible.
But when we're talking about something that small, it's easy then to see how, maybe, one layer of cloth over your face maybe wouldn't be sufficient to protect from a virus, or, protect particles and stuff moving in and out, because it's almost like a mesh, and this stuff just passes right through it because it's so fine. But then when you start adding in extra layers of cloth, then you can start to actually kind of increase the chance that those tiniest little particles are going to hit something else instead of pass right through.
So you mentioned earlier that you had the paper in December that you thought was done.
Yeah.
Did that paper get published?
No. So yeah, we were writing it up, and
you have to rewrite the entire thing now.
No, it was actually really pretty straightforward. So the paper was done, and then as we were getting ready to submit it, then this whole SARS-2 came out, and then it was basically another couple of weeks to finish up that bit of the story and put it in. So no, actually, we didn't need to change much at all. It was not right timing, but it coincided right when we were getting ready to publish.
Yeah. I think we have time for maybe one more question, and I'm really curious if you could answer any big question about viruses in the world. Where is your curiosity taking you? What is the question that, if technology wasn't a limitation, if field work or any of these challenges, resources that are so often challenges in research work were gone, what would be the big question you'd hope to be able to answer?
I guess if there's a common thread behind all of the viruses that we can exploit to shut them down. And I think there's some interesting data from some groups trying to get at this question of, is there some kind of broad approach we can take to prevent viral replication in a host without killing the host, of course. I guess that's the challenge.
That does seem a good challenge to work within those parameters.
But something that would go after all of the viruses rather than just one.
Yeah. Do you think that do all these different viruses have a commonality that is potentially?
Other than that they infect a cell, I think that's about where all the commonalities really start to fall apart. I mean, they're not all made out of the same things. They all infect different types of cells, use different receptors, look different.
Well, I did say it was a big question if you could remove a lot of the current limitations.
There's a lot to get at before we can go that far.
Absolutely. All right. Any final messages to our audience living here in this world with a global pandemic?
Yeah.
Excellent. And you just moved. I see the shelves behind you are still a little bit empty.
Yeah, this is my somewhat empty office.
What a time to change positions and start a new laboratory.
Yeah, it's different.
For sure. Thank you so much for joining us tonight and giving us a little bit of history and some backstory into these viruses, into how they cross species. And it's really interesting to see how they move from within species might be a great impact for the future and understanding and predicting where they might come from. So, so happy to have you here with us tonight. Happy to have all of you viewing with us tonight as well. Thank you so much for sharing your great questions, and really appreciate you joining us this evening. We hope that you've enjoyed tonight's event, and you can let us know your thoughts about it by filling out a short survey that we're posting right now to the YouTube chat. And that survey will also be sent out to everybody who's registered in advance. Feedback from you is very helpful as we plan our next programs, making sure that we are planning of interest to you. If you enjoyed tonight's talk, if you'd like to rewatch it, we've got a couple of different options for you. So first off, please consider a donation to PacSci to help make programming like this continue. And this talk will be saved on the PacSci YouTube page. So you can not only watch it again, but you can also share the link with friends and families. Be sure to sign up for our e-newsletter at pacsci.org/SITC. That's for Science in the City, to learn more about future programs. We do hope that you'll consider joining us for our next Science in the City event, which will be taking place on Tuesday, September 29th. That talk is called Calling BS, the Art of Skepticism in a Data-Driven World. So again, very timely talk as tonight's was. Thank you again so much for joining us. Michael, thank you very much for joining and sharing your expertise with us. Have a great night, everybody, and be sure to stay curious.